So, what is tandem mass spectrometer? It is an advanced analytical technique that involves multiple steps of mass spectrometry. In a simple terms, it is like a process where molecules are broken down in stages, with each stage giving us a detailed information about the molecule's structure. This makes it extremely useful in identifying and analyzing complex compounds. Tandem mass spectrometer is essentially a system that uses two or more mass analyzers in series. It helps in studying compounds in more detailed way compared to a single mass spectrometer. By definition, a tandem mass of spectrometer is a device that connects two or more mass analyzers in series. The purpose is it is mainly used to analyze a compound in clinical research and diagnostic. For example, it is important in studying protein and peptides, which are building blocks of life. It is also used for disease diagnostic and drug development. Now, let's talk about the main component of the system. There are four components. First, ion source. This is where a molecule are ionized to prepare them for analysis. Second, mass analyzer. This helps separate the ion based on their mass to charge ratio. Third, collision cell. This is where the ions are fragmented for deeper analysis. Fourth, detector. This capture and record the data. Alright everyone, now we talk about the different types of mass spectrometer. First, chlorophyll MS. This first type is called triple chlorophyll mass spectrometer. Imagine like it has three gates, one after another, that help sort molecules based on their mass. These gates, called chlorophyll, work together to select the specific molecules, break them apart, and then measure the pieces. This method is really great for measuring a precise amount of substance. For example, it is often used in a lab to check for specific chemical or even drug testing. Second, next we have arbitrary mass spectrometry. This one focus for accuracy. It is like having a super magnifying glass to measure the exact weight of ion, even down to the tiny differences. Arbitrary is particularly useful for studying things like protein or metabolites, which are small molecules in our body. Scientists love it because it gives a detailed result for research work. Now let's talk about matrix assisted laser desorption ionization time of flight. Quite a mouthful, right? Let me break it down for you. This method, a laser hits the sample and creates ions. This ion then fly through a tube and their time for flight is measured. What is cool is that perfect for identifying large molecules like proteins. It is fast and efficient, so it's often used in clinical diagnostic and microbiology. Fourth is the chlorophyll trap mass spectrometry. This combines two techniques, the chlorophyll method and a trapping system. Think of it like catching a molecule in a trap, breaking them apart and analyzing each pieces for its exact weight. It is great for giving a detailed and precise information about the complex molecules, which is a super helpful in drug research and chemical analysis. Now, let's break down the principle of tandem mass spectrometry. I will explain it in simple terms so it's easy to follow. Step 1 is ionization. Here's what happens. We take the sample we want to analyze and turn it into the ions. These are charged particles. Why do we do this? Because the mass spectrometer works by measuring the mass to charge ratio. So we need the sample in ion form to analyze in properly. Step 2 is mass spectrometer MS1. Once the sample is ionized, it goes into the first mass spectrometer MS1. Here, the ions are separated based on their mass to charge ratio. It is like a sorting people into a different group based on their height and weight. Each ion gets sorted so we know what we're dealing with. Step 3 is selection of ions. After the ions are separated, we move to ion selection. In this step, we choose a specific ion from the group for further analysis. Is it like picking one specific items from menu when you want to focus on it. Step 4 is fragmentation. The selected ions are sent into a special area called the collision cell. Here, they collide with gas molecule and break into smaller pieces. This step is more important because the fragments give us more information about the structure of the molecule. Step 5 is mass spectrometer 2. Now, these fragments move into a second mass spectrometer MS2. Here, the fragments are analyzed in detail, just like we did earlier in MS1, but this time is for smaller pieces. It helps us to dig up deeper into a molecule structure. Step 6 is detection of and spectrum generation. Finally, in the last step, the data from MS2 is collected by the detector. The detector generates a spectrum, which is basically a graph showing the abundance of different ions. The spectrum is like a fingerprint. It tells us exactly what compounds are in the sample and they move on its application. Then the mass spectrometry is widely used. Clinical diagnostic is for detecting disease like cancer or metabolic disorder. Pharmaceutical research is to analyze drug and their metabolites. Proteomic and genomic is to study the protein and DNA. Environmental science is to detect the pollutant.
While tandem mass spectrometry is a powerful analytical tool, it also comes with several limitations. I could list down as much as 9 several limitations, but I'm only going to focus on 6. 1. It requires technical expertise. This is because complex data analysis requires expertise in doing this MS. Other than that, the high cost. A machine can cost up to 100k and not all research center has the financial ability to purchase one. Other than that, mass accuracy and resolution. Mass accuracy and resolution depends on the type of mass analyzer that is being used. Other than that, limitation to ionizable compounds. The compounds can be ionized effectively by choosing a specific ionization method. Other than that, limited dynamic range. Samples that contain compounds of vast different concentration may lead to detection of low abundance. Lastly, ion suppression. This is because presence of salts buffers can suppress ionization and it can affect the sensitivity and accuracy of doing this method. Okay, now we will talk about how tandem mass spectrometry, how the operation of tandem MS. Tandem MS will go 5 stages, which is ionization, first mass analyzer, which is MS1, fragmentation, that is also known as collision cell, second mass analyzer, which is also known as MS2, and detection and data analysis. So, ionization is the process where the process begins with ionization, where the sample molecules are converted into charged particle ion, and this is done using the technique like ESI or MALDI, depending on the sample type. Secondly, it will go into MS1, which is also known as first mass analyzer. This process is to analyze filters and select ions of specific mass to charge ratio from the ionized sample, and these selected ions are known as precursor ions. And then the next stage is fragmentation, which also known as collision cell. These selected precursor ions are directed into a collision cell where they collide with neutral gas molecules like nitrogen or argon. This collision causes the ions to break apart into smaller pieces called fragment ions. This step is also known as collision-induced dissociation, which is also short form for CID, which reveals structural information about the molecule. Fourth step of this operation process is second mass analyzer, which is MS2. The fragmented ions are then passed into second mass analyzer, which separates them based on their unique mass to ratio values. This generates detailed fragmentation pattern or spectrum. And last of the operation process is detection and data analysis. The detector captures the fragmented ion data and generates a mass spectrum, which is a graph showing the mass to ratio values and their intensity. This spectrum provides crucial information about the structure, composition, or sequence of the original molecule. So this is just a visual representation of the tandem MS process. For the case study that we choose a case study that we have found, which is from the pharmaceutical industry. So they did was using the mass spectrometry in order to identify the metabolites in the new anti-inflammatory drug, which is trigasoline and ciprofloxacin. So in conclusion of doing this case study, that they had done was plasma. The result was in trigasoline, plasma concentration was a bit lower than the other samples, and this leads to a significant difference which should be calculated by pharmaceutical that can be calculated by for pharmaceutical parameters, which also makes into plasma clearance. And for ciprofloxacin, there was no significant difference. So this, in conclusion, by doing tandem MS provide a powerful tool and which can provide a detailed analysis of drug metabolism. It also allows precise identification and quantification of metabolites, which is very crucial in the drug development and in regulatory approval. In conclusion, tandem mass spectrometry is a highly versatile and powerful analytical technique tool, which can be widely used in various scientific fields such as proteomics, metabolomics, and pharmaceuticals, and such on. Its ability to select, fragment, and analyze ions in a very sequential manner allows for the precise structural elucidation and quantification of complex molecules. Despite its limitations such as high cost and technical expertise, the advantage such as high sensitivity and specificity cannot be erased. However, with the continuous advancement in MS technology and data analysis tools, which are very quite expanding, its application making it a very essential tool in the a very essential tool for the research industry. Thank you. So that is all for our presentation for today. Thank you.